Hello, this is the lecture for Chapter 16 on cell signaling. Now this is a really important chapter for many of you because it's going to be a lot of new information. So if you're going to be, say, taking the MCAT in the future, students have said that this section of cell bio is really helpful. And also if you've taken a lot of biology or zoology classes in the past, the introductory classes, typically cell signaling is not something that we get into. And it's also going to bring together a lot of different concepts that we've been learning so far in the semester. So we have 5 objectives for chapter 16. The first is to understand the general principles of cell signaling. So what is kind of the general path that a signal takes from outside the cell to inside the cell? Second, to be familiar with the different types of cell signaling. So there's a variety of different methods that cells can communicate with each other in the body, and you should know and be able to compare and contrast and provide examples of these different types. Third, you should know how intracellular molecules mediate signaling cascades. So we'll talk about what a signaling cascade is and what we mean when we're talking about intracellular molecules, to know why they are important and some examples of intracellular molecules. Next, we're going to talk about G protein coupled receptors. So this is a class of receptors, protein receptors, that are the beginning of kind of a signal coming from outside the cell to being transmitted in. And so you'll need to know all the different parts of that signaling pathway. And then last, we'll talk about enzyme-coupled receptors. This is another class of receptors. You should understand how they work and all of their components, what happens when things go wrong. So let's talk about our first objective, the general principles of cell signaling. So what I'd like you to do first, in class normally I would stop and have you guys brainstorm this together. Try to think of 5 ways that cell signaling is used in the body. Right? Think of 5 functions or reasons to have cell signaling. Right, hopefully you've been able to at least get 3. I've got a list here of some different ways that cell signaling is being used. First, during development of an organism, cell signaling is important for telling different cells what they need to become. It's important in the immune response. We've talked about macrophages before, we talked about antibodies, right? How the immune response works is dependent upon cell signaling. We've also talked about neurons and how they communicate, right? That is a type of cell signaling. Hormones are a way to regulate a lot of different processes in the body in a coordinated manner. And again, this is an extracellular signal that is turning on different pathways inside the cell. Cell migration. Certain cells have a property where they are attracted to different extracellular signals, and they'll crawl in the direction of that signal. And so that is another method that extracellular signals are being used. Drugs, really important, right? A lot of medications actually act on signaling pathways at various steps in these pathways. Muscle contraction. We've talked a little bit about muscle contraction when we learned about neurons and how they could stimulate a muscle to contract. Majority of your senses are all cell signaling, right? They're receiving some type of signal, some type of stimulus from the environment and turning that into some type of electrical response, right? All of our senses are connected to our nervous system. If a cell is going to divide, often it receives a signal from the surroundings that tell it to divide, such as, hey, it's time to grow this organ, or hey, this tissue's been damaged, we need to regenerate it. So cell proliferation is induced by extracellular signals. So these are just a few examples, and your book provides a variety of different examples of cell signaling and why it's important in the cell. So I like to think about this in comparison to how we as people communicate. So what are some methods that we use to communicate? So I would again encourage you to pause the video here and write down what are some examples of ways that we communicate as the human species. So we have face-to-face communication, we have phone call, text, TV, billboards, and now, of course, we've got YouTube, right, or different types of video conferencing. We have books, radio, newspaper, email, flyers, right? These are just a collection of a few different ways that we communicate. So think about these methods of communication. Which of these reach the most people, right? The most people at once. So maybe like a spam email could do that, right? TV would reach a lot of people, potentially, which would spread the fastest, right? So again, email is almost instantaneous, right? TV can be live as well. Some of these things take longer, right? Like a book or a newspaper to produce. Which of these can spread the farthest, right? Some of them are not very far, so face to face, you can't hear in the next room what people are saying, versus an email you can send to someone on the other side of the world. Which lasts the longest? Emails can be deleted in a second, right? But maybe a book, right, that's permanent on paper, that can last quite a long time. And so just like we have these different ways of communication in our world that are important for different types of messages, some of them need to be really fast, some of them need to last a long time, some need to be able to be refreshed or changed quite rapidly. The same goes for in the cell. Again, when we're talking about signal transduction or cell signaling, that's taking a signal from outside the cell, some type of molecule or some type of stimulus, and leading and using that to make changes inside the cell. So that's what they mean when they say transduce, right? It can changing forms. We're taking a stimulus from outside the cell and having some type of change inside of the cell. So think about some extracellular signals. Take a moment, pause the video, think about what are some types of signals that you currently know of. So here are a variety. This is not all of them. We've talked about some of them, such as hormones, such as testosterone, estrogen, neurotransmitters like acetylcholine, dopamine, epinephrine, drugs, right? Pretty much any drug that acts on a receptor. Cell-cell contact, so actual just touching, two cells touching has a potential to induce changes inside those cells. Cytokines are the signaling methods used in the immune system, so these get released by immune cells. Antibodies, we talked about how antibodies are this molecule outside of the cell and they can label foreign material to be digested. We have nitric oxide, so some gases act as signals. And we have growth factors, so these are a variety of molecules that are telling the cell to divide or to grow in size. So let's talk about the different types of cell signaling. So again, we talked about in human communication, you can have both long or short ranged, you can have temporary or long-term signals, and the same is true in the cell. So here we have four main classes of communication. First, we have endocrine, next paracrine, synaptic, and then finally contact dependent. So in an endocrine signaling method, a cell releases a signal and it gets put into the bloodstream. And since the bloodstream goes throughout the whole body to all of our tissues, that signal can be sent throughout the body and then bind to the appropriate target cell as long as it has the correct receptor for that signal. So hormones are an example of endocrine signaling. So cells produce a hormone, it gets released into the blood, and then that signal can act on a variety of different tissues in the body. Paracrine signaling is more short-ranged, so the signal is only going to reach cells that are nearby. Next, we have synaptic signaling. These signals can travel a long distance through the length of an individual cell, but the actual distance that the signal travels from one cell to the next is very short, right, to that synapse. And we talked about neurotransmitters being the molecules that mediate that interaction between two cells, two neurons. Then last, we have contact dependent. In this case, the cells have to actually touch each other, and once they touch each other, that tells the cells to do something. So there are some cells in culture in which their growth is contact dependent. So the cells will divide and divide and divide until they touch each other. And when they physically touch each other, that is a signal to both of those cells, all right, we need to stop dividing because otherwise we're going to get too cramped. That's contact dependent inhibition. So let's do an example problem. To maintain a local stimulus, paracrine signal molecules must be prevented from straying too far from their points of origin. So sometimes it's important for a signal to stay local and not to spread throughout the entire tissue or the whole body. What are some ways that you could help make sure that this signal stays local? So try and brainstorm some ideas of how this could be controlled. So most of these signals are short-lived. That is, the molecules that are released by the cells aren't very stable, and so they'll only last for a short period of time. That's kind of like printing a newspaper, right? They're on pretty cheap paper. They know that this is only going to be kept for about a day and then thrown away, versus a book or a statue, right? Something that's built to convey a message for a long time. The shorter something lasts, the harder it is going to be for that molecule to travel really far, right? In addition to maybe not being very stable, these molecules can also be degraded by enzymes in the surroundings. So there are enzymes out there that can break down these molecules. So again, they don't last very long, they can't travel very far. You could also have rapid uptake by neighboring cells, right? We've talked about this a little bit, a similar idea at the synapse, right? How serotonin reuptake inhibitors control how much of this neurotransmitter is in the synaptic cleft. There's a similar idea with paracrine signaling in that the amount of signal can be limited by being rapidly taken up by nearby cells. We haven't talked a lot about the extracellular matrix, but if you remember at the beginning of the semester, we learned that it kind of is like a web, that it contains a variety of different components, that it can be important for things like cell migration, for tissue structure, right, like collagen. So there are types of molecules in the extracellular matrix that can actually act like a sticky web and that can make it difficult for molecules to diffuse very far. All right, so let's try and take this idea of cell signaling being similar or comparative to human communication a little bit further. Right, so to help you really understand the similarities and differences between these different signaling methods, I encourage you to take a minute and to think about what are some ways that people communicate that could match. Right, so for example, of all those different communication mediums we described that people use, TV, books, newspaper, email, which of those best represents paracrine signaling or synaptic signaling, right? And there's no one correct, it's not like there's only one correct answer for each of these. So take a minute, pause the video, think about what you want to put for these problems. So I would say that a good example of paracrine signaling in human communication is the use of a flyer or a billboard. They are pretty short lasting, especially flyers, and they're very local, right? Flyers are put up in your neighborhood. A billboard is only going to be viewed by people that are close enough to read it. Synaptic signaling could be compared to telephone, especially back before we had satellites, right? Where you literally had to have telephone wires connecting people to connecting their telephones. Email is another example, right? These can travel very long distances. And also when you're having these when you're using telephone or email, often it is just kind of on a one-on-one basis, right? You're sending that email to one person or you're calling one person in specific. Autocrine signaling is not on this list, but it is another type of cell signaling. And this is basically auto self. So this would be a signal that turns around and acts on the cell that made that signal. So that could be compared to talking to yourself or journaling. Endocrine signaling. This also can travel a far distance, but it doesn't typically have one specific target like a synapse does. And so I think that TV is a good example, especially cable TV, right? Or radio. These messages can travel very far and they can reach more than one person. Lastly, the best comparison I can think of for contact-dependent signaling is a hug, right? You have to physically touch that other person to make them feel appreciated. So let's do a problem to delve more into the mechanisms of cell signaling. You treat HeLa cells with a hormone you are studying, but nothing happens. However, when you treat HEK cells, so this is a different type of cell, human embryonic kidney cells with the same hormone, they respond. How could this be? Why is it that one cell type responds to your to your hormone, but another cell type doesn't? Right, so take a moment and pause the video and think of what your answer could be to this problem. Now, all of these signaling methods that we've talked about utilize something called a receptor. So receptors are what receive the signal, kind of like you have to have a telephone to receive a phone call, or ears to hear someone talking to you, right? Cells that receive signals, those signals bind to a receptor. This is a protein that is attached to the surface of the cell facing outward. And these receptors can have varying degrees of selectivity as far as what signals they accept, right? So if you've ever tried to call someone or business and you accidentally use their fax number, it doesn't work, right? So in this example, it could be that HeLa cells do not have that hormone's receptor. So every different type of hormone or every different type of neurotransmitter has a specific receptor that will receive it. And so different cell types have different functions. They're going to have different types of receptors. So it could be that the HeLa cells just don't have that receptor for that hormone. So no matter how much hormone you dump in that dish, the cells will never respond to the signal. Let's do another problem. You are a behavioral neuroscientist, and you discovered a new species of mouse that is very tame and will not bite. Right? So other researchers want to know if its brain works the same way, right, as normal mice. So do the brain cells respond to neurotransmitters in the same way as the current mouse models, right? So you've got this mouse, it behaves differently. They want to know, how is its brain similar or different, specifically in terms of how it responds to neurotransmitters. And how could you test this? So take a minute, pause the video, and try to answer this question. We know that neurotransmitters have to bind to a receptor, right? And so there's a variety of different experiments that you could use to test this out. There's not one right answer. So first, you could treat this new mouse species with a neurotransmitter, and see how it responds behaviorally, right? You could treat it and then look at the brain like slices. You could take those cells out of the brain, culture them in a dish, and treat the cells in the dish with the neurotransmitter and watch how they respond. Right? But that is only one half of the story, right? For every neurotransmitter type, there has to be a receptor. So the other half of the experiments that you would need to do would be to determine if there are receptors for all the different types of neurotransmitters present in the original mouse. So you could do tissue labeling for all the different types of receptors, acetylcholine receptors, epinephrine receptors, dopamine receptors, right? And you could look and see, do all the same cell types have those receptors, as well as do all the same cell types make those neurotransmitters. So again, you could label the mouse brain tissue for neurotransmitters. And you've done cell labeling before, right? So you did immunocytochemistry. That was labeling proteins, cells in a dish. If you were looking at slices of tissue, it's the same technique, just slightly modified, called immunohistochemistry. So histo has to do with tissue, right, slices. So you could use immunolabeling to identify where those neurotransmitters are present. You could also use labeling to look not only for the neurotransmitters, but for their receptors as well. Again, the important concept that I'm trying to get at here is that every receptor is selective in what it responds to, right? So a receptor will not bind to every type of signaling molecule out there. And so this is really important, right? Because if you are a certain cell and you have a certain job to do, it might not be good to be hearing signals that other cells need to hear. You need to focus. So how do we go from taking this outside stimulus and turning it into some type of change inside the cell? So what we use to describe that change, or at least the proteins that cause that change, is the word effector. So effector proteins are the proteins that are going to cause the end result of that signal. Let's look at an example. So acetylcholine is a neurotransmitter, and we've talked about it before, how it would be released by neurons that are interacting with muscles and will bind to muscle cells to tell them to contract. For a muscle to contract, it has to use a variety of different cytoskeletal proteins, such as actin, troponin, and myosin, right? So those are the kind of the final destination targets of the signaling pathway, because these are the proteins that are involved in the actual contraction of the muscle. Now, the same signal can have different effects depending on which cell it is targeting. So acetylcholine, for example, in muscle cells, it will cause contraction, but in salivary gland cells, it will cause secretion of basically spit, right, saliva. And so it's a really nice, efficient mechanism that you can have the same signal travel to different cell types and cause kind of a coordinated response. So in this case, on a salivary gland cell, the effector protein is aquaporin, because the end effect that we're looking for in the salivary glands is not contraction, it is secretion of saliva. And saliva can be secreted through these pores called aquaporins. Right? So we have the same signal, but when it attaches to different cell types, it has different downstream effects. Here we have 3 different examples. We've got heart pacemaker cells, salivary gland cells, and skeletal muscle cells that all have different responses to the exact same signal, acetylcholine. Now, cells don't only receive one signal at a time or one type of signal, and so they also have to integrate a variety of stimuli and determine what the cell should do next, right? So I have kind of a big family. Sometimes I come home and like 3 different people are talking to me at the same time, and I've forgotten that I used to know how to handle that. And so I have to say, slow down, one thing at a time. But these cells are receiving signals from a variety of different sources, and they have to combine all of those and decide what the next best action is. So it could be a set of signals may encourage the cell to survive, to divide, to mature and differentiate, or in the absence of signals, many cells will just undergo apoptosis and die. Now, the response of a cell, the response inside of a cell to a stimulus can be either fast or slow. So let's think about the different types of end effects that you could see. So for example, you could have a gene be turned on and a protein be made. You could have an enzyme be turned on or off. You could have a change in the membrane potential of a cell. A cell could be induced to divide or to undergo endocytosis, right? Some of these can happen really quickly. So turning a protein on or off could be as simple as phosphorylation or dephosphorylation. That can happen really quickly. Membrane potential. We've talked about how this voltage change can travel very, very rapidly down the surface of the cell. Changes in gene expression take a bit longer. You have to have the DNA be transcribed and then translated, and then that protein has to be modified and sent where it needs to go. So that would be a slower response compared to an electrical signal, an action potential. Secretion, again, depends. If that cell, if this is regulated secretion, that could be quick if the cell is ready to go and all of those molecules to be secreted are waiting in vesicles at the surface of the cell. And cell division, that I would say is probably medium because there are changes in expression of genes that needs to occur, but then there's also proteins that are ready to go that just need to be phosphorylated, for example. So we've got, again, just summarizing here the different types of responses to cell signaling. Now we've talked about receptors that are at the surface of the cell. You can also have cell signaling in which the signal does not stop at the surface of the cell, and it doesn't bind the receptor on the surface of the cell. Some signals can actually just travel right through the plasma membrane. In that case, some of these signals, they are going to bind to a receptor that rather than being on the outside of the cell is somewhere located inside the cell, or they may just skip a receptor and act directly on an enzyme, turn that enzyme off or turn it on. Right? So what would allow a signal to travel through the plasma membrane to not have to use a receptor or a channel? So hopefully you've remembered that things that are small and nonpolar can diffuse through the plasma membrane relatively quickly. And so here we have some examples, such as nitric oxide, right? That is a small molecule. It is nonpolar. It can travel through the membrane very quickly. Now, some of these signals that travel through the cell membrane will go directly to the nucleus and change gene expression. They'll tell a gene to be turned on or turned off. So in that case, there are receptors in the cytosol or at the nucleus, which then are acting as transcription factors. So it's important for you to remember what transcription factors are. We've talked about them as proteins that bind to DNA in the promoter region, and they tell a gene whether it's going to be transcribed or not. So whether or not messenger RNA is going to be made, and ultimately if protein levels are going to change. Here we have a cartoon showing an example. Cortisol is a signal that can diffuse through the plasma membrane, and it binds to a receptor protein inside the cell. So this is called a nuclear receptor protein, and it guides this signal into the nucleus, and then it can bind to a promoter region and turn on transcription of a target gene. Another example in which a signal is traveling into the cell and skipping the cell membrane or skipping receptors at the cell membrane is nitric oxide. So in this case, it does not bind to a receptor inside the cell or outside of the cell. It acts directly on an enzyme. In our example with nitric oxide, we can see that a cell, an endothelial cell, can release nitric oxide. In this case, it's releasing it in response to acetylcholine binding. So you can kind of disregard the acetylcholine portion of this. What I want you to notice is that this nitric oxide, this signal, can travel across the plasma membrane. It does not need to bind to a receptor. And its target is an enzyme, guanylyl cyclase. What this ultimately leads to is relaxation of smooth muscle cell. And the example that we have here, you can see smooth muscle cell is in direct contact with endothelial cells. So the cells that are releasing the nitric oxide are very close to the cells they are acting on. And in this case, acetylcholine is going to cause release of this gas. It's going to spread from the endothelial cells to the smooth muscle cells and cause them to release. So when this is working on a blood vessel, when the smooth muscle cells contract, that causes the lumen, that hole, to be smaller. And when the muscles relax, that makes the blood vessel larger. So this can be a way for cells, tissues to regulate blood flow and blood pressure, right? And this is actually how Viagra works. So Viagra acts on this guanylyl cyclase signaling pathway, so in the smooth muscle cells, to relax those smooth muscle cells that surround blood vessels. Let's talk about signaling cascades. Right? So what happens once a signal binds to a receptor? Now again, we said that cell surface receptors, they are selective. They are binding to signals that are outside of the cell and relaying that information inside of the cell to have a targeted effect. So some ways that people talk about this this process of relaying a message as a molecular relay race, like you're passing the baton from one person to the next in a relay race. It's also called a signaling pathway or a signaling cascade. So if you think of a cascade like a waterfall, you have bouncing, splashing, causing another flow of water over here, kind of bouncing from rock to rock. Or flow, right? Cascade flowing. So these are all mediated, the signaling pathway or cascade, whatever you want to call it, this is mediated by intracellular signaling molecules. So if you had a relay race, each person in the relay is like an intracellular signaling molecule. And here we have just a generic example of a receptor and a signaling pathway. So you can see there's an extracellular signaling molecule that binds to its matching receptor, and then the receptor protein acts on molecules inside the cell in this relay, kind of this cascade or pathway. So those green molecules there are representing our intracellular signaling molecules. Where they end up and what they finally act upon is the effector proteins. These can be things such as enzymes, cytoskeletal protein, or transcription factors, as we've talked about before. So we're going to spend some time talking about each of these different parts. The primary transduction, so that's the initial receiving of the signal. The relay, so the message being transmitted from one molecule to the next. We then have a step called amplification, where instead of being a one-to-one relay, now you have a speaker and you are broadcasting that information. So it's amplifying, right? Strengthening the signal. Then the cell inside the cell has to respond to all the different types of signals it's receiving, integrate those, decide what to do, and then tell the various target proteins in the cell what changes they need to make. So, based on that generic pathway that we saw, what would be one way that you could regulate the intracellular signaling pathway? So if you wanted to stop this pathway, stop a signal from working, for example, or if you wanted to enhance a cell's response to a certain signal, what could you do? We've talked about pathways before. We talked about glycolysis, for example, and we talked about this technique or this process called feedback regulation. That is where a downstream product can act on a previous upstream protein. So you can have positive or negative feedback. So in positive feedback, the downstream product is upregulating or enhancing a previous protein in that pathway. And in negative feedback, it's saying, slow down, right? So there are signals that can turn proteins in the cell on and off. So a lot of these intracellular signaling molecules can be turned on and off, right? So if you wanted to regulate the signaling pathway at the intracellular level, you could turn any of those proteins that are in that chain on or off. And turning off is important, just as important as turning on. For example, what if you could never tell your cells to stop making saliva after receiving a signal from acetylcholine? That would not be good. We'd all be big drooling messes. So we've talked about proteins being turned on and off, right? And so a lot of the similar methods that we've talked about before about enzymes being turned on and off, those are the methods that are used in signaling cascades in response to extracellular signals. So we have two main types that you should focus on for this chapter. First of all, phosphorylation, and this one we've talked about before, right? So phosphorylating a protein, an enzyme, in some cases it can activate the enzyme. In other cases it can deactivate the enzyme. It just depends on the situation. But phosphorylation is like a switch, right? Another type that is a little bit newer to you is GTP binding. So you've heard of ATP, adenosine triphosphate, adenine triphosphate. GTP is a very similar molecule, except instead of adenine, it's got guanine, right? And so it's a molecule with a nitrogenous base that has 3 phosphate groups attached to it. And when it has 3, it's called GTP, triphosphate. And when it has 2, it's GDP, diphosphate. And so in this example, we can see that when GTP is bound to this enzyme, that makes the enzyme be activated. And when it is bound to GDP, diphosphate, the enzyme is inactive. Right, so again, phosphorylation or GTP binding are 2 of the main ways that these intracellular signaling proteins are turned on and off. A little bit more about the details of this. So only certain residues or certain amino acids in a protein, in an enzyme, actually get phosphorylated. It's not like you can attach a phosphate group to any amino acid. So interestingly, there are only 3, serine, threonine, or tyrosine. Those are the 3 amino acids in a protein that can be phosphorylated. And an interesting and important detail about GTP binding is how this GTP gets turned off or dephosphorylated, right? So in this example, we can see that when GTP binds, it activates the cell. And what happened? GDP was removed and GTP was added. Then within a certain amount of time, this GTP got hydrolyzed. It lost a phosphate group, and now it is GDP, and this enzyme is inactive. So what's causing it to be hydrolyzed? In fact, the enzyme itself that it is bound to can hydrolyze its own GTP. And so when it does that, it's called a GTPase. And don't worry, we'll talk more about how this works. So the way I like to think about this is like wearing shoes, right? So if you have wear shoes for a certain amount of time, eventually they'll get worn down, they'll be kind of gross, and you'll need to get a new pair, right? So in this case, your new pair of shoes is like GTP, and this GTPase, that's like me, right? The more I wear my shoes, the more worn down they get. So after a certain period of time, and it just depends on what type of enzyme it is, it will hydrolyze, it will break off one of those phosphate groups. And now instead of being bound to GTP, it's bound to GDP, right? It's the same molecule, it just has one less phosphate group. Now you may say, why not just rephosphorylate this GDP? Can we just add a phosphate group back onto it again and now the protein is active? Unfortunately, no. So it's kind of like for most of us, we don't try to fix our shoes, we just throw them away and we get a new pair, right? So it's throwing away this GDP and it's putting on a new pair of shoes. It's putting on this new GTP. So again, when we say something is a GTPase, it inactivates itself. It's kind of like it's got a timer on it, and that at a certain point, that GTP that's bound to it, it's going to break off one of those phosphate groups and turn it into GDP. Again, you cannot add that third phosphate group back onto the protein to reactivate the enzyme. Instead, you have to get rid of the GDP and replace it with a new molecule of GTP. So there are three types of receptors: ion channel coupled, G protein coupled, and enzyme coupled. So ion channel coupled, if you think about those words, ion channel, that has to do with membrane potential. So these are channels that are responding to changes in the voltage of the cell. G protein coupled and enzyme coupled. So there's receptors that work via something called a G protein. And there's other receptors that send their signals inside of the cell by enzymes. And so you should know these three different types and how they work. And again, the same signal can act on multiple types of receptors. So you can have the same signal bind to a G protein on one cell, or a G protein coupled receptor on the surface of one cell, or bind to an enzyme coupled receptor on the surface of another cell. Here we have an example in which we have acetylcholine, right, binding to a G protein coupled receptor. That is what's shown in A. And then in C here, this is an example of an ion channel coupled. So in this case, when this molecule binds, the receptor is also an ion channel. And so we've talked about this before, right? When we talked about how neurotransmitters can send signals, right? So you have a ligand that's binding to a receptor, but this receptor is also a channel, which is pretty cool. And so when the ligand binds, the channel opens up and allows ions to flood in or out of the cell and change the membrane potential. Let's talk about G protein coupled receptors in more detail. like to watch a video with you guys to introduce this section. Right, so what did I do? I went on YouTube and I tried to find the scariest video that would not actually make people angry at me. All right, so some of you watching this, especially if you have fear of heights, you may have needed to close your eyes for a second. You may have felt your heart rate increase a little bit, all right? So we are talking about, in this example, the hormone adrenaline, right? So things like going up on top of skyscrapers or skydiving, right? These different activities, these people, we call them adrenaline junkies. Adrenaline is a hormone that actually its effects are mediated by these G protein coupled receptors. And so when you think about adrenaline and what responses people have to adrenaline, we know some of them, such as increased heart rate, but that's not the only effect of adrenaline. We also can experience faster breathing, dilated pupils, and in fact changes in digestion, right? So it's cool because if you are in a situation normally where you need to respond, protect yourself, you're going to want increased heart rate, faster breathing, more oxygen to your cells, to your muscle cells, pupils dilating so you can see and sense any danger, changes in digestion so that you're not focusing all your body's energy on digesting food, but instead that energy is being directed to muscles so that you can get ready to run or to fight. So all of these different responses that your body needs to have at one time can all be mediated by one molecule, adrenaline. So in here, you may be wondering why do I have a picture of Sylvester Stallone? Well, this is a very famous scene from the movie Rocky in which he's getting ready for the big fight. He's getting all pumped up and motivated. And a lot of times we play this song, maybe in a basketball game, right, to get the crowd excited. So I'm not going to sing it for you. But if you don't know what song I'm talking about, you can go ahead and Google it. It's called Eye of the Tiger. All right. And same thing, that song is supposed to get you kind of your hormones going, your adrenaline, get you motivated to, in this place, beat your partner, right? And of course, also the one that I've been talking about, but I forgot that it was on this list, prepare your muscles, right, for that fight response. So again, adrenaline and many other signals are using G protein coupled receptors. These are receptors that will, once they receive a signal, are going to mediate the effects of that signal by a protein called a G protein. And because this is such a long phrase, a term, we just for short call them GPCRs, which is really not much shorter either, right? So this is the largest family of receptors. They are responsible for majority of our sensory abilities, and a variety of signals, a variety of proteins, molecules, not just hormones, will act on these receptors. So in addition to hormones, we can have local mediators, so paracrine molecules, neurotransmitters can bind GPCRs as well. And like we talked about before, they can have multiple functions, such as our senses, smell, taste, vision, hearing. They're important for immune cell migration, so for those immune cells to go where an infection is. We talked about the adrenaline response. Importantly, half of our drugs are working on GPCRs and their downstream targets. Right? So very important signaling pathway. Researchers have learned a lot about these pathways and have been able to modify them to help cure and heal different. So we're going to talk about all the different parts from starting at the top where the ligand binds all the way down to an effector protein for G protein coupled receptors. I said that they are able to relay their signal to inside the cell using G proteins. And the reason these are called G proteins is because they bind and are activated by GTP, right? Guanosine triphosphate. In this diagram, we have an example of a GPCR, a receptor that has bound to a signal, and that has caused activation of a G protein, and it is activated when it is bound to GTP. So binding of the signal molecule has led to GTP binding to the G protein and activating it. And once activated, this G protein can leave the receptor and travel to other parts of the cell. Looking up closer at this G protein coupled receptor, it is a 7-transmembrane domain protein. So you can see here that it spans the membrane 7 times. We just talked about transmembrane proteins, right? So where do you think that this protein was synthesized? Hopefully you said by ribosomes on the surface of the endoplasmic reticulum, the rough ER. So as this protein was made, it had a signal sequence on the terminal end that told the cell that this protein was going to be inserted into the endoplasmic reticulum. And it had, after that signal sequence, it had 7 stop transfer sequences. 7 transmembrane regions. These are hydrophobic regions. And that directed the translocator to have this protein spanning the membrane 7 times. We've talked about secondary structure of proteins and that commonly alpha helices are used for insertion of the plasma membrane. And so this is a great example. So it's likely that of all of these alpha helices, that the side chains that are sticking out, they're not shown just for simplicity, that all of these side chains in this area have hydrophobic properties. Whereas the side chains that are on these parts of the proteins that are unstructured and that are facing outside of the cell or inside of the cell, they probably have a lot of polar side chains. So a G protein is composed of 3 subunits that can attach and detach: alpha, beta, and gamma. You should know that the alpha subunit is the unit that GTP binds to. In this example, we have a signaling molecule binding to our receptor, a 7-transmembrane domain protein. And when it binds, it causes a conformational change in the receptor protein. So its structure shifts and changes conformation. All right. What that does is that also causes a change in the shape of the G protein, specifically the alpha subunit. And so this protein currently, before the signal binds, is inactive, and it becomes activated by binding of that signal molecule. So what happens is that as the structure of that alpha subunit changes, its binding pocket, where GDP or GTP binds, also changes. When it changes shape, it is able to release GDP and to bind GTP, GTP being the activating form. All right, so here it has this conformational change, right? And so now it's able to release GDP and bind to GTP and be activated. So this is a great example of structure determining function, right? Now, once the G protein is activated, that is what stimulates the subunits to split apart. And what we usually see is the alpha subunit separates, and then the beta and gamma subunits stay together. So this trimeric protein splits into two parts. Let's look at this in a little more detail. Here we have a cartoon demonstrating the structure of GTP. Guanosine is in black and the triphosphate, each of those phosphates is an orange circle. And so the activating form is GTP. When it loses one phosphate, it becomes GDP, and that is the inactive form. So normally, the G protein is all in its trimeric or 3 subunit state, and it's bound to the receptor, right? It's inactive. We know it's inactive because it has GDP, diphosphate, attached to it. When a signal binds to the outside of the cell on that transmembrane protein, on that receptor, that causes a chain effect of structural alterations that allows the GDP to be released from the alpha subunit here and to be exchanged for GTP, the activating form of this molecule. Once activated, this will cause the alpha subunit to separate, to disassociate from the other half of the G protein, and then it can have other downstream effects, which we'll talk about in a little bit. Now, this alpha subunit, the G protein, is a GTPase. We talked about that on a few earlier slides. What that means is that there's basically an inherent timer in this protein, so that eventually it's going to hydrolyze that GTP. It's going to break off one of those phosphates, and that protein molecule turns into GDP, which is the inactive form. So basically, it's kind of inactivating itself after a certain period of time. And you can see, right, it's lost this phosphate group. What will happen then, since the alpha subunit is inactive, it's going to rejoin the beta and gamma subunits at the surface of the cell attached to that G protein receptor. So what does that alpha subunit do? What does that G protein do when it's activated? So the first place it will go is to other proteins that are attached to the membrane on the intracellular side. So here we have a target protein shown in brown, and when the G protein gets activated by GTP, the alpha subunit travels, just kind of shuttles back and forth along the cell membrane on the inside surface of the cell until it reaches that target protein, and it can act on that target protein. When it binds, it's going to slightly change the conformation of that protein, which can activate it or inactivate it. So in this example, an active G protein is causing activation of a target when it binds. Now, this is a very important point. The response of this signal of a GPCR is regulated by how long this GTPase, this G protein stays active. How long does it take for this GTP to inactivate itself? So the longer this G protein is active, the longer it can be acting on its target. In addition, right, the amount of time that it is bound to this target regulates how long this target is responsive. So you should know those two methods of targeting or of controlling a protein response. Again, the G protein is a GTPase. It will inactivate itself after a certain period of time, and it does that by breaking or hydrolyzing the third phosphate group in GTP, and so now it becomes GDP, which is the inactive form. Now you can see that it's inactivated. When it's inactive, the G protein can no longer interact with its target protein, and instead it will go and complex with the rest of the G protein, the beta and the gamma subunits. So we talked earlier about how turning a signal off is just as important as turning a signal on. And here we're going to look at an example of some diseases in which Dysregulation of signaling pathways can have a catastrophic effect. So being able to shut a signal off is an opportunity for control, which is a good thing. Anytime you have a switch, right, you can have a risk of mishap. A switch can get left on or left off. So cholera is an infection of the small intestine that leads to diarrhea and vomiting and dehydration. And so in this case, it's a bacterial infection that can commonly be found in unclean drinking water. And cholera, the bacteria actually affects its effects are mediated by GPCRs, G protein coupled receptors. Here we have a demonstration again, just from earlier. We have, in a normal situation, a signal molecule binds to a cell in the small intestine, binds to a receptor on the surface of that cell, activating the G protein. And in this case, the G protein is targeting an enzyme called adenylyl cyclase. What the cholera toxin does is it modifies the G protein alpha subunit. What it does is it causes the G protein to be unable to inactivate itself, right? So the GTPase function of this G protein no longer works, and so it's never going to switch out, it's never going to convert GTP to GDP. What that means is that this G protein will remain constantly active and will be able to constantly activate its target, adenylyl cyclase. And in the case of the small intestine, the function of adenylyl cyclase is to regulate the flow of chloride and water into the gut. And so if you have dysregulation of ions and water in your intestines, that is how you can have things like vomiting, diarrhea, dehydration. And if not properly treated, can be lethal. So this is a great example of how that really shows whether or not you understand the signaling pathway. So if you can understand why modification of this G protein is leading to overactivation of adenylyl cyclase, then you've got this pathway down. Let's look at another example you can use to study whooping cough or pertussis. In this case, it's infecting cells of the lung, and it's causing coughing, runny nose, sneezing. It's a poor child with whooping cough. Okay. So let's look at how GPR signaling works in the lung. So in these lung cells, it's a little bit different of a situation to begin in a healthy lung. So in the lung cells, what this activated G protein does is when it binds to a target protein, rather than activating the target, it turns it off. So when a protein signal molecule binds to the transmembrane protein here, the GPCR, it activates the G protein just like normal. The alpha subunit travels to a target protein at the surface of the cell. But when it binds, rather than turning the enzyme on, it turns it off. So normally we want this enzyme turned off. At a certain point, the GTP will be broken down to GDP. The G protein alpha subunit will be inactivated. It will not be able to attach to the target protein anymore. And so when it releases the target protein, that reactivates the target. So in the lung, G protein inactivates adenylyl cyclase. So when you have, so normally adenylyl cyclase being off in lung cells is a good thing. However, the pertussis toxin prevents the G protein from being activated. What it does is it affects the protein, the alpha subunit here, and so it is unable to switch out GBP for GTP. So what does that mean for that alpha subunit? It means that this protein is going to be inactive. all right? It cannot go and travel over to this target protein because it's inactive. And in this case, that means that the protein is just going to stay active for a very long time. And in the lung, that is not a good thing. So active adrenalyl cyclase in the lung will stimulate coughing. Let's do a problem to test your understanding of GPCRs, right? What would be the consequences of a mutation in the G protein that causes its affinity for GDP to be reduced without changing its affinity for GTP? So this mutation is making the alpha subunit to bind more loosely to GDP, right? So what happens if the alpha subunit doesn't bind tightly to GDP? That means it's going to be able to lose that GDP very quickly and more frequently. What will happen as soon as that GDP is gone? It will be replaced with a GTP. So what this is going to do is it's going to enhance the reactivation of this alpha subunit. As soon as it gets activated, or as soon as it converts that GTP to GDP, as soon as it gets inactivated, it will get reactivated very quickly. Right? What this ultimately will do is will lead to overactivation of the target. So in this example, we see that when the active G protein is bound to the target enzyme, that it promotes enzyme activity. And so what that would mean is that if the G protein is more frequently being activated, the target protein is also more frequently being activated. So let's compare this with the effects of the cholera toxin, our first example. The first example also resulted in overactivation of the target enzyme. It was by a different mechanism. And so it's important to note that because there are multiple steps in this pathway, there are multiple points of regulation, right? Just like in the game Telephone, with every person you add in that game, the more that message has the potential to get screwed up. So you should be able to take this pathway and say, what happens to the signal downstream effect if I change this or if I change that? Again, an important thing that I want to emphasize is that with this cell signaling by GPCRs, that you can have the same signal causing a variety of effects depending upon the cell target, right? And there's a lot of variety. There's kind of mix and match of these different components. For example, the ligands that bind to GPCRs can range from things like light, so smell, taste molecules. They could be neurotransmitters like epinephrine or acetylcholine. They could be hormones like testosterone, right? Could be medications. So there's a variety of different ligands that can bind to GPCRs. And then there's also a variety of different effectors. So far we've talked about adrenalyl cyclase. We've also talked about ion channels. And in the case of adenylyl cyclase, we even saw some variety where some signals lead to activation and others lead to inactivation. Other effectors can be phospholipase C. We also have this PI3 kinase. If you look at the G protein itself, there are variations of G proteins, different subtypes. And then also what happens downstream of, say, adrenalyl cyclase can vary depending on the cell type. So let's look more closely at the example of an ion channel being regulated by GPCRs. We're going to look at heartbeat. So heartbeat is controlled by muscles, and these muscles are told to contract by two sets of nerves. One set of nerves speeds up heart rate, and another set slows down heart rate. And so focusing on how heart rate is slowed down, these nerves will release acetylcholine, right? So this is a neurotransmitter, and it binds to the pacemaker cells of the heart, and the receptor is a GPCR. So here we have a cartoon of a cell, a cardiac pacemaker cell in the heart. It has a receptor for acetylcholine. When acetylcholine binds, it will activate this G protein. And what the G protein will do is it will lead to opening of a potassium channel. I don't know if you can hear that. Sheriff's coming by. Anyways, so the G protein will lead to activation of this potassium channel. And in fact, in this example, it is not the alpha subunit, but the beta gamma subunit, which is activating it. So hopefully you still remember what happens when a potassium channel opens in a cell. Is potassium more highly concentrated inside the cell or outside of the cell? It's more concentrated inside of the cell, which means that it will rush out. What will this do to the membrane potential? If you have a lot of positive ions leaving the cell, that means that the membrane potential, the inside of the cell, is going to become more negative. The more negative the membrane potential is, the harder it is to induce an action potential. Because for an action potential to happen, the membrane potential has to rise. So this is called hyperpolarization. The harder it is to activate the pacemaker cells, the more infrequently they are going to tell the heart to contract. So by acetylcholine binding to this specific cell type, it is causing slowing down of heart rate. So we've talked about ligands, we've talked about the GPCR itself, and kind of the G protein, and a target enzyme we talked about was adenylyl cyclase. But what happens to that target protein right after that? So that target protein at the membrane is not necessarily the end of the message. So right now we've had kind of a one-to-one message. One ligand binds to one receptor, activates one G protein, which activates one target. The second messengers are an opportunity to amplify that signal, to magnify the signal further downstream. So one of these targets that we've talked about of G proteins is adenylyl cyclase. And the other enzyme that can be a target of G proteins is called phospholipase C. Both of these enzymes are going to produce second messengers. Adenylyl cyclase and phospholipase C, they are going to release these small intracellular molecules. So they are going to send molecules down deeper into the cell that aren't attached to the membrane, and these are called second messengers. So the second messenger of adenylyl cyclase is called cyclic adenosine monophosphate, or cyclic AMP. CAMP is another way people describe it. And the second messenger of phospholipase C, there's three: inositol triphosphate, diacylglycerol, and calcium. So we're going to go over these in more detail, but you should know what these secondary messengers are. Again, the duration of a signal's response to a signal is controlled by how long the G protein is activated and how long the G protein is bound to the target. The longer the G protein is bound to adenylyl cyclase, the more cyclic AMP it can make, right? Here you can see the next step in this GPCR signaling. Activation of a GPCR leads to GTP binding to that G protein. The alpha subunit separates, travels to its target, which in this case is adenylyl cyclase, and activation of adenylyl cyclase results in the synthesis of cyclic AMP from ATP. Cyclic AMP can be made very quickly, and so it's a great response for signals when the response needs to be really fast. The more cyclic AMP, the more of the signal is being sent inside of the cell. If you get rid of the cyclic AMP, you stop the signal. So some enzymes or some extracellular signaling molecules that cause production of cyclic AMP in the cell are adrenaline, glucagon, and adrenocorticotropic hormone. Adrenaline, for example, it's going to act on a variety of tissues, heart, skeletal muscle, fat, and depending on the cell type, it has different responses. But the first part of that pathway involving adenylyl cyclase and making cyclic AMP is the same. Here we have a really cool experiment where scientists measured cyclic AMP levels inside of the cell in response to a signal. So what happens in this cell is it is treated with serotonin, or 5-HT, and serotonin causes production of cyclic AMP in the cell. And higher levels of cyclic AMP are shown as brighter yellow and red and purple, and lower levels are more like blue. You can also see on here the different amounts of time: 19 seconds, 49, 109, 10 minutes, and 15. So within 20 seconds, you can see a rapid increase in the amount of cyclic AMP, and this signal is gone within 10 minutes. So it's a pretty fast signal. It's temporary, but it's very rapid. So what happens if you make cyclic AMP? What does cyclic AMP do? Cyclic AMP acts on a target called protein kinase A, or cyclic AMP dependent protein kinase A. I know, super creative, right? So cyclic AMP molecules can bind and activate pKa, protein kinase A. Based on its name, what do you think pKa does? Hopefully you said it phosphorylates things, right? Because it is a kinase. And so pKa can have two downstream effects. One, it can turn on enzymes, and two, it can turn on gene expression. So let's take a minute to stop and take a step back and think about back at the beginning of the lecture when we discussed different types of communication, right? So compare each steps of this signaling cascade to the following types of human communication: telephone, conversation, or radio announcement. Take a moment to solve this problem. So the first interaction I have listed as telephone. Adrenaline binding to a GPCR. We have one molecule binding to one target protein. So that's like a telephone conversation between two people. Next, we have one G protein interacting with one adenylyl cyclase protein. Again, one-to-one, it's like a telephone conversation. Now, when we talk about second messengers, this is an opportunity to amplify, like a megaphone, to ramp up the signal. And so the step in which adenylyl cyclase makes cyclic AMP is more like a radio announcement, because now, rather than just talking to one person, you're talking to everybody in that county, for example. Then, lastly, we have telephone, right? One cyclic AMP molecule activating one pKa molecule. But because of that amplification step, step 3, rather than having one adrenaline molecule resulting in activation of one pKa, we can have one adrenaline molecule resulting in the activation, in this example, of 9 pKa proteins, right? 9 enzyme molecules being activated. So the signal has been amplified. So again, pKa can do two things. It can activate enzymes. So in the example of skeletal muscle, pKa will act on an enzyme called phosphorylase kinase. Right? So pKa is phosphorylating this enzyme and activating it. Active phosphorylase kinase acts on an enzyme called glycogen phosphorylase. You've heard of glycogen before, right? So it's activating enzymes that are important for metabolism in skeletal muscle. So we talked in that table about adrenaline working via adenylyl cyclase and cyclic AMP. And what it is doing is it is causing breakdown of glycogen. And we know that glycogen is a long-term storage method for sugar. And if you are needing to run or to fight, right, adrenaline response got you going, your muscles need to be ready to contract a lot. They need a lot of energy. And so your body's going to start breaking down stores of glycogen so that it is ready to go. Pretty neat, huh? So we talked about cyclic AMP being a rapid signaling method, but I'd like you to take a moment to understand why it must be rapidly broken down. So we know that it must be made very quickly, but why should the levels also drop really quickly? Take a minute, pause the video, think about this problem. So it's important for cyclic AMP levels to stay low in the cell so that when a signal is received, we can have a big jump, right, in the levels of cyclic AMP. Right? If there's already a lot and you add more, the cell may not respond very much to that signal. In addition to activating enzymes, adenylyl cyclase can also lead to induction of gene expression. Again, here we're looking at an example of adrenaline, and this time cyclic AMP, when it activates pKa, does not lead to activation of enzymes, but rather expression of genes. pKa can enter the nucleus when it becomes activated, and it can phosphorylate transcription factors. So just like enzymes can be turned on or off by phosphorylation, the activity of transcription factors can be regulated by phosphorylation as well. So in this case, a transcription factor will not bind and promote a gene being transcribed until it gets phosphorylated. So only upon signaling of adrenaline will this specific gene get turned on. So we've talked about GPCR signaling. We've talked about adenylyl cyclase being one of those intracellular targets, one of those enzymes that the G protein acts on. Another major enzyme that G proteins activate is phospholipase C. So you should understand not only how G proteins can activate adenylyl cyclase, but also how they can activate phospholipase C. Phospholipase C is a little bit more complicated. What it does, once activated, is it breaks apart a protein, a lipid, that is at the membrane. We have this molecule, inositol phospholipid, that is hanging out at the plasma membrane. And it can be broken by phospholipase C. Lipase, fats, phospholipids, kind of see the connection? So when this inositol phospholipid gets cleaved by phospholipase C, the two resulting pieces are diacylglycerol, or DAG, DAG, and inositol 1,4,5 triphosphate, gotta love your biochemistry, or IP3. Right? So phospholipase C can lead to cleavage of the inositol phospholipid, whose resultant parts are diacylglycerol and IP3. You'll note that diacylglycerol will stay attached to the membrane, whereas IP3 is released and now it can do stuff further inside the cell. Phospholipase C is going to ultimately lead to a rise in calcium inside the cell. And you should know that normally calcium is kept at very low levels inside the cell. Do you remember where stores of calcium are kept? So there's a certain organelle that stores calcium so that it is ready to be released inside the cell. So the smooth endoplasmic reticulum contains a high concentration of calcium. So in order to induce calcium response in the cell, it doesn't necessarily have to let calcium in from surroundings. It has a little closet stashed full of calcium that it can open whenever it needs to. And that closet is the endoplasmic reticulum. So you can imagine that since diacylglycerol is stuck in the membrane, that's not the one that's going to be releasing calcium from the endoplasmic reticulum. Instead, that IP3, that piece of the inositol phospholipid that breaks off, can travel further into the cell and can open a calcium channel on the surface of the endoplasmic reticulum. Right, so the G protein, upon activation, travels to phospholipase C and activates it. This enzyme's function is to cleave inositol phospholipid into two parts, diacylglycerol and IP3. IP3 can then travel to the endoplasmic reticulum and open calcium channels, causing a release of calcium into the cell. Now, calcium is a very important signaling molecule, so the cell is very careful to regulate calcium levels, and there are certain proteins that will respond to calcium. So let's look at some examples in cells and tissues that respond to phospholipase C activation. Here we have acetylcholine, vasopressin, and thrombin. These work in a variety of different tissues and have different responses. However, they all are using phospholipase C activation and cleavage of that inositol phospholipid to mediate this variety of results. Let's do a problem, right? I encourage you to take a minute to work on this as well, pause the video. There's more than one right answer to this problem. So, if you wanted to prove that phospholipase C cleaves inositol phospholipid, and that cleavage leads to an increase in calcium inside the cell, you really have a two-part experiment. Because first you have to prove that phospholipase C can break this molecule in half, and you also have to prove that when this molecule gets broken in half, that it leads to activation of this calcium, opening of this calcium channel. You also have to show that this opening of the endoplasmic reticulum is connected to phospholipase C. So the first step that you could take is you could use a drug to inactivate phospholipase C. So this drug, you could inactivate it, and then you could look at levels of calcium in the cell, as well as levels of this inositol phospholipid, whether or not it's getting cleaved or not. If phospholipase C is important, when you inactivate it, no matter how many signaling molecules you release into that dish, the calcium levels will not rise, and the IP3 will not be broken apart. If phospholipase C is not involved, you would add this signal and you would see a rise in calcium. Another step you could take to really show this connection is you could create an inositol phospholipid that could not be broken apart. And so if the inositol phospholipid is unable to be broken apart, and you treat the cells with this signal, and you see that calcium levels do not rise, that's telling you that this inositol phospholipid is important for the signaling mechanism. If you treat the cells with the signal and calcium levels rise despite the fact that the inositol phospholipid is not cleaved, then you know that some other mechanism is involved. GPR signaling is really great for quick responses. So things, again, we've talked about adrenaline, that's a very fast response. Mouth watering at a smell, that's pretty quick. Vision, cell migration, these are all mediated by GPCRs. Here we have a cool video that demonstrates how fast GPCRs can work. In this video, they release a signaling molecule from the end of this pipette. And you can see that it is an attractive molecule, and the cell will migrate towards it.
These human neutrophils, taken from the blood of a graduate student, are mobile cells that will quickly migrate to sites of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source. When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
I don't understand the bird call at the end, but it's a pretty great video. And it's amazing to see how fast that cell responds, because we know everything that's going on now. We know that there's a G protein, that there's some type of enzyme at the membrane that's being activated, that secondary messengers are being produced. And also think about all the rearrangement of the cytoskeleton that must be happening for that cell to migrate. So this is super fast. So we're going to have another question, all right? How is it that different cells can respond in different ways to exactly the same signaling molecule, even when they have identical receptors? So you have two different cell types, and they both bind to the same ligand, the same signal, yet they have completely different responses. How is that possible? This is possible because different cells have different G proteins and target proteins, so those proteins that the G proteins act upon. If you remember, I showed you this picture earlier. You can see that even if you have the same ligand binding to the GPCR, you can have different subclasses of G proteins and different effectors, depending upon the cell type. The last type of receptor that you should know is enzyme-coupled receptors. So enzyme-coupled receptors, as their name implies, they have enzymatic activity. So they don't interact with G proteins inside the cell, but instead, the portion of the protein that is facing the inside of the cell is acting as an enzyme itself. Alternatively, it could form a complex with another protein at the cell membrane, which is an enzyme. Now, enzyme-coupled receptors are typically responding to signals such as growth factors, local signals, and they tend to have a slower response time than GPCRs. One very common example of an enzyme-coupled receptor is called a receptor tyrosine kinase, or RTK. So this is not the only enzyme-coupled receptor out there, but it's a very well-known enzyme-coupled receptor that we're going to focus on in our examples. So receptor tyrosine kinase, for short, is called RTK. So you can guess what it does by its name. The intracellular facing side of this receptor protein is going to act as a kinase. It will phosphorylate proteins that it interacts with, and on those proteins, it is going to phosphorylate the amino acid tyrosine. Now, the importance of enzyme-coupled receptors, such as receptor tyrosine kinases, can be seen in cancer. And so a lot of drugs are being designed against these signaling pathways because they are involved in promoting things like growth and proliferation of cells, as well as differentiation or maturation of cell types. And we know that all of these different processes, when dysregulated, can cause cancer. The structure and function of receptor tyrosine kinases differs from GPCRs in a few ways. First of all, there is only one transmembrane domain, so it only spans the membrane once. And they often will dimerize, so two receptors will come close to each other and together bind a ligand. And when they're brought together, what's happening is those intracellular regions of the proteins that are enzymes can start acting, and they can act on each other. In this case, we said that they are kinases, so they will phosphorylate the respective receptor and cause activation of both of these receptors. You can see in this example that the ligand is also in the form of a dimer, and that helps to recruit two of these receptors close together. Now they are close enough that they can phosphorylate each other's tyrosines, and they both become activated. Now, phosphorylation of these receptors kind of sends a signal to the rest of the cell that proteins can come and bind. So they're acting kind of like a docking region that is only available, it's only open for binding when it's been phosphorylated. So you can see in this cartoon, we've got a variety of other intracellular signaling molecules that are now being recruited to the active receptor tyrosine kinase. And some of these molecules, in turn, because they are close to this kinase, this receptor tyrosine kinase, they can also become phosphorylated in order to activate their properties. Again, looking back at our generic description or cartoon of a signaling molecule, I want you to notice here a scaffold, right? So scaffolds are proteins that act as like a docking site. Again, they're just a region to help facilitate the formation of complexes of proteins. And so some of these proteins that are recruited to the receptor tyrosine kinase, their main job is just to help associate other proteins in this area. Now, receptor tyrosine kinases can activate a variety of signals at the same time, resulting in a variety of things happening at once. So in this example, you can see binding of one signaling molecule is actually activating a variety of effector proteins in the cell. Let's think again. Now that we know kind of some of the main steps of this signaling cascade, what can we do to regulate it? We know that tyrosines are being phosphorylated to activate them. And so if you could remove those phosphate groups, you could inactivate those intracellular signaling molecules. And so we know enzymes called phosphatases remove phosphate groups. An alternative method to control the response of a cell would be just to remove the receptor altogether. And one way the cell can do that is by endocytosis. So this region of the cell membrane would form a little pocket, a bud facing inside of the cell, containing that region of the membrane that has the receptor, and it would ultimately be endocytosed and digested. Endocytosis of receptors is a very important method of regulating a cell's response. So what happens once this receptor tyrosine kinase gets activated? Now there are a wide variety of pathways that can be activated by receptor tyrosine kinases. However, there are some components, some pieces of this pathway that are consistent across all the different types. And so the first component we're going to talk about is called RAS. This is a GTP binding protein involved in the MAP kinase signaling cascade. And we have our familiar phospholipase C. So most receptor tyrosine kinases, which are a type of enzyme-coupled receptors, will bind this protein called RAS. RAS is a GTPase, so it will bind to GTP. And this RAS protein associates with the cell membrane. And it's able to do this because it has a lipid tail. So this would be a post-translational modification, a covalent attachment of a fatty acid to that RAS protein. That fatty acid, because it is hydrophobic, can insert into the plasma membrane, but it can also move around. And that is the same way that G proteins are able to shuttle back and forth across the membrane. Now RAS, since it has this GTP binding capability, it has this switch activation that we've talked about before. Here is RAS in its inactive form. It is associated with the plasma membrane by that lipid tail, and it right now when it's inactive is bound to GDP. When a signal binds to a receptor tyrosine kinase, this causes activation of the enzyme of the tyrosine kinase, recruitment of additional proteins at this area. Now they've docked here and they're in close association with RAS, which is also at the cell membrane. One of the proteins that binds to the receptor tyrosine kinase is called RAS-GEF. And this is guanosine exchange factor, or GTP exchange factor. It is important to have these enzymes because they help with reactivation of these GTP binding proteins. So the GDP will get removed. We talked about like getting rid of old shoes and buying a new pair. That is mediated by this RAS-GEF, GTP exchange factor. Now that the RAS protein is bound to GTP as it is active, it can have further downstream effects in the cell. And we mentioned that one of its downstream targets is the MAP kinase signaling cascade. So the MAP kinase signaling cascade, a kinase signaling cascade, kinases, so phosphorylation of multiple proteins. So one protein phosphorylates another, that activates that target protein, which can then phosphorylate another third protein. These tend to be serine-threonine kinases. So they are adding these phosphate groups to serine or threonine amino acids on their target proteins. So this is really like a domino or relay effect. And it's ultimately going to end up causing changes in the nucleus. So the final target is mitogen activated protein kinase. So that's where the MAP comes from. You may have heard of the word mitogen before. Mitogen has to do with growth. And so MAP kinase is going to help mediate signals that tell the cell to divide. Now, proteins in the MAP kinase signaling cascade have dumb names. All right, so we said the final target is MAP kinase. So to get to MAP kinase, the RAS protein is going to activate MAP kinase, which then phosphorylates and activates MAP kinase, which then ultimately will phosphorylate and activate our target MAP kinase, which is pretty silly. Now this MAP kinase, as I said, it can act in the nucleus, it can change gene expression levels, it can also, since it is a kinase, it can regulate protein activity as well. So again, we're really seeing this domino effect. We've got, once we get to the final MAP kinase, we have a variety of effector proteins that are being targeted. So RAS, again, RAS has been activated by an RTK, receptor tyrosine kinase signaling mechanism, that's causing ultimate activation of MAP kinase, and it's promoting things like cell growth and proliferation. And because of that, RAS is known to be associated with cancer, since it's regulating things like cell growth and proliferation. And in fact, a third of human cancers have mutant RAS, right? They're over expressing these signals for the cell to proliferate, right? It's an activating mutation. So something's happening so that this RAS protein is always activating the MAP signaling cascade when you have too much proliferation. So we've talked about the important common factor RAS in RTK signaling pathways. Another common component of receptor tyrosine kinases is the modification of inositol phospholipids. So receptor tyrosine kinases, again, we've talked about this emphasis of their role in cell growth and survival. Some of the signaling molecules can be insulin-like growth factor molecules, or IGF. Right? A growth factor is a type of signal that we talked about at the beginning of this chapter. So receptor tyrosine kinases, they will activate each other by phosphorylation, and then they will lead to activation of other enzymes that are recruited to the cell membrane. One of those is phosphoinositide 3 kinase. So this is another type of kinase that's being activated, right? And what it does is it phosphorylates inositol phospholipids. Right? We've talked about inositol phospholipids before. Here we have an example where instead of being modulated by a G protein signaling pathway, it's being affected by receptor tyrosine kinases. In this situation, the inositol phospholipid is getting phosphorylated. Right? So we have a signal molecule such as a growth factor binding to receptor tyrosine kinases, causing dimerization. That causes activation of the receptor, and then they are able to phosphorylate and recruit enzymes to this region, acting as a scaffold. And one of those proteins that gets recruited is this PI3 kinase. PI3 kinase will phosphorylate an inositol phospholipid. What does this inositol phospholipid do? It then can act as a second docking site. And so another enzyme, particular to this inositol phospholipid, will be recruited at the cell surface. One example of an intracellular signaling protein that is mediated by this phosphorylated inositol phospholipid is AKT. AKT is a super important signaling protein, and it is a kinase as well, again promoting cell growth and survival. In this case, what AKT does is rather than activating enzymes or turning on gene expression, it is turning off proteins. So any proteins whose job is to induce cell death, AKT will shut those down. Again, this is going to promote growth and survival in the cell. So in this case, the phosphorylation of inositol phospholipid due to receptor tyrosine kinases is leading to activation of AKT, and the downstream effect is that it's inactivating proteins and it's preventing cell death. How it does that is by a molecule called TOR, right? So we've got receptor tyrosine kinase activating PI3 kinase, PI3 kinase phosphorylating inositol phospholipid, allowing the recruitment of AKT. And the way AKT is able to promote cell growth, we said, is that it inhibits proteins that lead to cell death. So it does that, again, by this TOR protein. TOR is going to inhibit degradation of proteins in the cell and also enhance production of proteins. So this is beneficial for helping a cell to grow and to survive. Again, since we're dealing with cell growth and survival, that you can expect that these components of the cell signaling pathway are going to be involved in cancer. For example, the cancer drug rapamycin inactivates TOR. So that is going to promote cell death. Now, I want to pause for a moment and talk about just these pathways in general. We've talked about ion-coupled receptors, we've talked about GPCRs, and we've talked about enzyme-coupled receptors. And these aren't necessarily always acting separately on their own, right? For example, we talked about inositol phospholipids being regulated by both GPCRs and by receptor tyrosine kinases. And so I don't expect you to memorize this figure, but what I want you to see is that things are not independent. There is a fair amount of interaction between these pathways. And that is what helps these cells to integrate a variety of signals from the surroundings. Now, let's talk about some cell contact mediated signaling, right? So one example is Notch signaling. This is a great example of signaling that is contact dependent. This is important during neural development. What we can see here is we've got a tissue of cells, and over time they're maturing in two different types, but you can see there's a pattern, right? It's very evenly spaced out, these two different cell types. And this is mediated by contact-dependent signaling. So how does this actually work? We have two cells that have to touch each other in order to induce signaling in each of those cells. And so just like other signaling methods use receptors and ligands, there are receptors and signaling molecules here. One receptor is going to be bound to the surface of one cell, and the signal, instead of being free-floating in the extracellular matrix, is attached to the surface of another cell. So only when those cells get close enough will the signal bind to the receptor. Once that signal binds to the receptor, this Notch signaling method has a way to quickly respond. And so what it does is the intracellular portion of this receptor will actually break off and travel to the nucleus. And it will turn on a specific set of genes acting as a transcription factor. And so what happens is when these two cells contact, one cell gets a certain signal that says, all right, become this, and the other cell gets a signal that says, okay, become that. And so it allows them to become two different cell types, and that promotes this pattern formation of, like, say, every other cell being a different type, which is pretty cool. Now, we talked about integration of multiple signals, all right? And so I want to talk a little bit about how that can work. And an important method for doing this is a protein kinase network. So we've just really brushed the surface of cell signaling here. So as you saw on one of those previous slides, there's a lot of connections. In this example cartoon, you can see that binding of some signals inhibits the signal pathways of others, right? And ultimately what this is doing is regulating whether kinases turn on or off and activate a target protein. And so what I really want to emphasize with this slide is first that protein kinase networks can help to integrate all these signals, but also that it's really complicated and it's more of a web than a chain. And so signaling pathways are a very active area of research. Anytime a scientist discovers a new phenomena, they want to know, how exactly does this work, right? That's why I got into research. I want to know exactly how things work. And so often scientists, once they discover something, they will then want to see every step in that signaling pathway. So that's it for this lecture. Again, these are the 5 objectives that you should know for our 4th and final exam.